So this is the class for Bio 461, Molecular Biology. And my name is Shiraz Mujtaba. I am Associate Professor in the Department of Biology at Medgar Rivers College, CUNY. And I have been teaching Bio 461 since a couple of years. My educational background is that I'm a PhD, and my PhD is in the area of HIV, molecular biology, and immunology. And after finishing my PhD, I did my postdoctoral training at Mount Sinai School of Medicine in the area of transcription regulation, which now is called as a field of epigenetics. So at Mount Sinai, I was there as a postdoc. I grew up to become independent faculty. I had a research lab, I had own grants. And after 13 years at Mount Sinai, I moved to CUNY for my passion for teaching. So at Mount Sinai, I was doing 24 hours research. So I wanted a little bit of change in my career. So I joined CUNY, and since then, since 2014, I am in CUNY. And since then, I'm teaching Bio 211, that is Biotech and Society. I teach Bio 461. I also teach during spring Bio 472, which is molecular biotechnology. And I also teach senior seminar, but that is Bio 499. So I look forward to having a wonderful semester. I hope you learn, and I hope that I also learn from your feedback. And needless to say, we moved into a virtual world for literally everything, from shopping to teaching to many things. So, society changes, and we should be able to change with time and as per the requirement. So that's called as flexibility. So asynchronous is also a form of flexibility, where we don't have a live lecture, face-to-face lecture, but it depends upon your convenience, and which is very good thing. But only thing that I see, because molecular biology is a subject which goes in detail, so you might have questions, and which should be addressed. So make sure that you attend office hours. So on the Facebook of the syllabus, you have my name, my location on the campus, but we are not on the campus. Then you have my email address. This is my email address. And most importantly, I want you to focus on my virtual live office hours. And I have given this on 3 different days of a week. On Tuesday from 12 to 12:30 p.m., on Wednesday from 4 to 4:30 p.m., and Thursdays from 6 to 6:30 p.m. My intention is that everybody gets to ask a question and attend the live office hours, right? So with this brief introduction of me and this course, I am going into the details of this syllabus, right? So one of the important component of syllabus is that course overview, right? And so there are a few things that I want to say before start. that when you are attending a live session, number one, you should mute yourself, right? Because there are more than one individuals in this live session. Second, uh, it's up to you, but it's strongly recommended by CUNY that you don't use video, uh, you know, don't use your picture ID, and for attending these class, right? You can just link, you can attend. I have already emailed you the link, which is a recurring link. I will not be sending new link every week, but that's the same link that you can use. That's towards your convenience. And so number one, be muted. You don't have to use your video unless you want, but it's strongly recommended by CUNY. Don't put your picture, don't come on digital media via videos, and we all know why. So keep that in mind. Now going into the course overview, this course is geared towards teaching you fundamentals of biology or molecular biology. So what do I mean by, this means by fundamental is that we all know that the central rule of most of the living organism is DNA serves as a template for RNA synthesis, and RNA serves as a template for synthesis of proteins, right? So this is, so from DNA to RNA, this is called as transcription, and from RNA to protein, it is called as translation. This is the scenario with most of the living organisms, but there are very salient examples which are exception to this rule. And one of the most well-known exception is HIV, which has its genome as RNA. So this HIV don't follow the conventional rule. The conventional rule is that from DNA to RNA to protein, but in HIV, it is from RNA to DNA, but this DNA which comes from RNA, this is called as complementary DNA, cDNA. And this then cDNA would again give rise to RNA by transcription, and RNA would serve as a template for the synthesis of proteins. Right? So now another example of exception to this this rule of central dogma is what we are facing today, that is called with COVID-19. So this is an RNA virus, which is not like HIV. HIV is also RNA virus, but COVID-19 is also RNA virus. With HIV, you have a very important protein called as reverse transcriptase that converts HIV RNA genome into cDNA. In COVID-19, what you have is also RNA, but this virus doesn't have reverse transcriptase like HIV. This has RNA dependent RNA polymerase. So think about it, that how much your knowledge is practically useful in current situation. And you might be knowing also that a recent one of the medicines that has been approved for some cases of COVID-19, some severe cases, that medicine is called as remdesivir, right? This has been, although this is not very effective in all situations, but it could be tried. And this medicine, this medicine is effective effective against RNA, DNA, RNA polymerase. So think about, so these are this is the major step, but there are exceptions to this course, to this rule of central dogma of biology. And one is HIV, and the second could be COVID-19. And different, why these are important? Because these viruses are there in the environment. They could transmit. It could be from animals, it could be from humans, it could be from plants. So, so this course is geared towards teaching you this pathway, how this RNA, how this DNA is converted into RNA, and how RNA serves as a template for the synthesis of protein. That's the major message of this course. If I have to define in one sentence, it is to teach you the molecular basis for transcription and translation. So let's go back to the syllabus. So that's the course here, the central dogma. And also there's a lab component to this course, which I'm sure you're aware and you might be enrolled into that. So now, one of the most important, now what is a syllabus? A syllabus, I always share with my students that a syllabus is just like a business plan, which tells you that what are you going to learn in this class, what I am going to teach, and how I am going to teach. That's another aspect, but it will list the course objective. So learning objective of a course is the major nucleus of a course, student learning objectives. So, it's my duty to fulfill that student learning objectives or student learning outcomes. Now, it depends upon you how much you gain. And so I will go through the course objective or student learning outcomes, which is listed here. So there are there are 11 points. I will go one by one, and I'm going in detail because you will get an idea what is coming in this next few weeks or during this semester. So design experiments to prove that DNA is the genetic material. So remember, all these discoveries about biology didn't happen in one day. It happened simultaneously with the development of technology. Some of the very important concepts were given just on the basis of observation, and then scientists followed that. What is the example for that is the Mendelian law. Mendel's law described the theory of or laws of inheritance, law of dominance, law of segregation. But those laws or those hypotheses at the time of Mendel was based upon observation of pea plants. And when he proposed his hypothesis, it became a law because scientists could see that inheritance in human diseases also. For example, in sex-linked diseases, they follow Mendelian pattern of inheritance. So that's why Mendel is still called as father of genetics. But scientists, this opened a total new area of genetics, inheritance of characters. We all receive characters from our parents, but these characters don't come in air. It comes as a chromosomes, right? And chromosome has DNA, and DNA has alphabets, right? I'm talking to you, let's say in English, and English, I'm using words. Words are made up of alphabets, sentences. So I'm communicating with you in English with using alphabets, words, sentences, and all those things. So DNA also communicates with alphabets, but DNA has only 4 alphabets. ATGC, adenine, thymine, guanine, and cytosine. So what I was referring you to, Mendel's work. So Mendel's work opened the whole field of genetics, and then people wanted to focus on chromosomes. When people discovered chromosomes, they wanted to look at DNA. They wanted to look at the chemical composition of DNA, how DNA is transmitted. So everything happened phase by phase. One information led to new questions. New questions led to designing of new experiments. Those experiments gave new data. Again, that gave rise to new questions. So that's how scientific discovery happens. That you keep asking questions based upon the data that is emerging. So one of the first objective is what experiments were designed to establish that DNA is still, or DNA is a genetic material. Now, just just a couple of minutes back, I said that there is an exception because not every living organism has DNA as its genetic material. There are viruses which are in other words, non-living, but when they infect cells, they become living, they have genetic material as RNA. Now, point number 2, compare general organization of prokaryotic and eukaryotic genomes, right? So, biology is very diverse field, very diverse area of education. Why I say that? Because the living organisms are so vast. Living organisms could be distinguished on the basis of number of cells, right? An organism could be unicellular like bacteria. Certain eukaryotes are unicellular. So there can be two types depending upon the number of cells, unicellular and multicellular. The second division could be based upon the presence or absence of chloroplasts. If a chloroplast is present, then it falls into plants. If chloroplast is absent, it goes into animal kingdom. So chloroplast can divide the whole living organism into two types. Then use of oxygen can divide living organisms into aerobic, that utilizes oxygen for metabolism or for living, or non-aerobic, which doesn't utilize oxygen. Fourth could be presence or absence of nucleus. So if a nucleus is present, it is eukaryotes. If a nucleus is absent, it is prokaryotes. Now, this course could easily focus on humans, right? But it also focuses equally on bacteria, prokaryotes. Why? Because most of the discovery of molecular biology, it happened using bacteria as model system. So it was not possible to use human cells at that time or in all the lab, but bacteria were available to do the experiments. So these are laboratory strains in which scientists, and as I go into chapter 1 next week, you will see what I meant. So knowledge of eukaryotes and prokaryotes, you might have learned that in your Bio 101, Bio 201, or 211, but we are going to revisit the differences between bacteria and human cells, particularly the genome, in much more detail. So let's go to the next points on the learning objective. Third, compare the processes of replication, transcription, translation in prokaryotes and eukaryotes. So remember that in a bacterial cell, the DNA is in the cytoplasm. In bacterial cells, the DNA is in cytoplasm. But in eukaryotic cells, there is a special organelle inside the cells, eukaryotic cell. This is called as nucleus. And within this nucleus, you have chromosomes, right? So a human cell is much more complex than bacterial cells. Bacteria is simple. So all the processes in bacteria, replication, transcription, and translation happens in cytoplasm. Whereas in human cells or eukaryotic cells, replication and transcription, they occur inside the nucleus, and translation occurs inside the cytoplasm. Why? Because the ribosomes, they are present in cytoplasm. So a DNA that give rise to RNA, this RNA has to travel to the cytoplasm. And in the cytoplasm, there is ribosomes. On the surface of ribosome, translation takes place. So I gave you just a very surface difference between the prokaryotes and eukaryotes with respect to transcription, replication, and translation. But as I go into this chapter, which we talk about in chapter, I believe, 3rd, 4th, and 5th. So that will be about transcription, translation, and regulation of transcription. So that took care of 3rd point. 4th is explain the flow of genetic information based upon central dogma. So when you integrate the knowledge of transcription, translation, gene regulation, then you understand how complex or how interesting this system is and how information flows. It is not haphazard, it is very planned. And one thing you'll find very interesting is that I always share this, that we talk about DNA, we talk about cells, but if you look at very carefully, what is DNA? DNA is made up of nucleotide. A nucleotide is made up of sugar, phosphate, and nitrogenous bases. What are these? These are chemical molecules made up of atoms such as carbon, hydrogen, oxygen, so on and so forth. So knowledge of chemistry, although this is a molecular biology class, but I always tell my student, revisit your fundamental knowledge of chemistry. Look at carbon atom, look at hydrogen atom, look how they form bonds. For example, in DNA, right? So I can say DNA is a double helical molecule and it is a storehouse for information, but that's not simple. It is double helix. DNA is a double helix. But if you look at the backbone of DNA is sugar phosphate. And what is the bond? Phosphodiester bond. So phosphodiester bond, right, connects sugar and phosphate. So you must know how a phosphodiester bond is formed, what atoms are involved, what molecules are involved, why it is called as diester, why not it is called as monoester. So yes, biology is what we are dealing with, but the basis of biology is very much chemistry. So keep your knowledge of chemistry very handy. It will help you in long term. I always give this advice to all my students. Chemistry is very important because all these proteins, carbohydrate, what are those? Those are chemical molecules made up of atoms that enters into molecular interaction to make complex macromolecules, right? So keep your knowledge of chemistry very handy. It is very important that you don't lose sight of chemistry. It's extremely important for long term. So let's go back to the syllabus. Then mutations. Mutations are very important. So let's say so what is the central dogma of biology? Central dogma is DNA transcribes into RNA and RNA translates into proteins. Right? Now, if the DNA is bad, right? If the DNA is bad, then RNA will also be bad, right? And if RNA is bad, then proteins are also bad. And what will this result into? This will result into disease, right? So changes in the sequence of nucleotides in DNA will impact the sequence of RNA, and RNA will impact protein, and that is what you see in one of the most well-known disease called as sickle cell anemia, right? Where a hemoglobin that should be a hemoglobin should be spherical shape, and it is very important. Hemoglobin is very important protein because it has red blood cells. They have a protein called as hemoglobin, right? It has a protein called as hemoglobin. Now, if there is a mutation in the sickle cell gene, what happens is that this spherical RBC becomes sickle shape. It's no more spherical. So this is a drastic transformation in the structure of red blood cells. And that happens because of hemoglobin. And so this sickle cell hemoglobin cannot carry oxygen, cannot carry carbon dioxide. So just one change in nucleotide. So what happens, in what I mean by change is there is a charged amino acid. It, by mutation, it becomes non-charged or non-polar, right? So this impacts the spherical structure of the protein. The protein just collapses, and a huge impact. Now, so these most mutations are very, very difficult if it happens in humans during birth, but there are exceptions also that mutations are good. For example, let's say there are some proteins. If there is a mutation in those proteins, then uh, a person is less likely to get infected by HIV. So these are good mutations. These are mutations that are present on one of the proteins of T cells that HIV would like to infect. But if these mutations are present, then HIV may not infect that successfully. So these mutations are now being used to make stem cells that can be used to replace the disease cells in AIDS patients or HIV positive patients. So mutations are bad. Conventionally, we always think mutation could lead to diseases, but there are mutations which are good also. So we will study about mutations. Then we will look at compare homologous and non-homologous recombination. Now the word recombination is very important. Recombination is responsible for variation between two generations. So we get genes from both the ends. We get from our mother, we get from our father, and then there is a, nobody knows the rule, but there is an arbitrary mixing of genes. And so we all look similar to our parents, but very different. So recombination is essence for diversity. And this homologous versus non-homologous chromosomes, you must have studied. It happens in meiosis, but it happens during DNA repair also. Not only in meiosis, in meiosis 1 in particular, prophase 1 or in prophase 1, I believe it is in diplotin, where crossing over occurs. But this crossing overs is with regard to DNA repair mechanisms. When one concept also I always give my students is that very, very fundamental, is that we know that if I ask you how many chromosomes are present in our cells, Everybody knows that there are 46 chromosomes. But what I try to revise with my students, that they're not 46, but there are 23 pairs. Means that there are two sets of 23 chromosomes deployed. Now these chromosomes in meiosis, they change part, and that changes uh, the, the sequence of DNA, and that gives rise to diversity. So that's what we are going to study in homologous and non-homologous replication. Then gene regulation, point number 7, compare the general organization of a gene and gene regulation. So think about New York City, right? It's such a densely populated city. At any point of time, there's traffic on the streets. But if the traffic is uncontrolled, then it will give rise to many accidents, many problems, right? But these traffic lights, this traffic is controlled by traffic lights, right? And if you have, let's say, streets and avenues, they're crossing at each other at right angle, then again, there are traffic lights. The traffic lights, what does it do? Traffic lights, it regulates the traffic of the city to avoid accidents, for good for citizenship, right? Citizens, right? So likewise, the traffic in our cell is from DNA to RNA, RNA to proteins, and this traffic has to be regulated because there are so many processes that depends upon this traffic. So this process is regulated, which is called as gene regulation. So that we will study in point 7 and 8 of the learning objectives. And then we go into the applied part of what we learned, the last chapter, which is on DNA technology, right? So that will be experimental manipulation of gene, how to make knockout, and what is PCR, what is siRNA, so on and so forth. So this course starts from the discovery of DNA, and then it goes into the knowledge of chromosomes and DNA. Then it goes into replication, transcription, translation, regulation of genes, mutations, recombinations, and then DNA technology. So it's this course goes very vertical. So when I was teaching other courses, in that course, one of the courses that I taught, there you learn about virus, bacteria, fungi, different kinds of plants, then you come back to human system. So it's a it has a very big landscape, right? Going from viruses to humans. But this course just focuses on the structure and functions of DNA, which is replication, transcription, translation, regulation, so on and so forth. So this is a very focused course on molecular biology, and this is very important course. You can relate it to disease processes, you can relate it to microbiology, you can relate it to cell biology. So it's the nucleus of many courses of biology. You know. So what's the textbook? Textbook is Essential Cell Biology by Alberts, and the publisher is Garland Science. Right? So that is a very good book. They have this book has very good figures and so on and so forth.
I think with the new.
Now I am trying to discuss with you the grading policy. So there will be 3 tests, test 1, test 2, and test 3. I'll tell you when the test 1 will be given when I go more down the syllabus. There will be quizzes after 1 or 2 chapters. So expect it to be 3 to 4 or more quizzes, particularly in online system. In asynchronous system, there are students who may not attend office hours because of good reasons. And I may not, so it's very important that I track the academic performance, so that I don't want a student to come to me at the end of the semester that say, so quizzes are important to know for you also and for me also to know how we are doing in this new system. So we went online just in March. Now this is a full-fledged semester that is online. So it's very important that I track and you know also how you are doing. And then if you need any advice, suggestions, and then we can work together. So quizzes are very important. Then assignments. So generally I give, let's say if this is a face-to-face session in the classroom, then I give class assignments to be done in the class in group. But that's not possible here in asynchronous system. So you get assignments as homework. Then another thing very important in online system is discussion board. Discussion board helps us to be connected, right? So every week you will get a topic to write on discussion board. These topics will not be from molecular biology, but they will be open-ended questions which would be a reflection or extension of molecular biology. So 3 tests, then 3 to 4 quizzes, assignments, and discussion board. And these are the various breakups of the contribution of each of these learning or assessments. Okay. Now, before I go into the course structure and the pedagogical part of it, I would like to pause here and I would like to take questions from you. Okay. So, any question? So one of the students asked me that whether the exams will be multiple choice questions or open-ended. Now, most of the exams will be multiple choice questions, but the assignments will be short answer questions and discussion board will be open-ended questions. I keep multiple choice questions because to keep it very straightforward and simple, because what I'm noticing is that everybody is using different system. Everybody has a very different speed of internet. So I try to keep exams multiple choice questions, but assignments and discussion board, that could be short question or essay type questions. So any more questions? Okay, if there are no questions, then I'm proceeding with the remaining part of the syllabus. So first chapter will be history and overview, which will teach you about how DNA was discovered as a genetic material, how DNA was discovered as a double helix, what are the structure and functions of DNA, and how the genetic information flow is in our cells. So this chapter is very basic. It goes into the discovery of DNA into detail, but it is it lays the foundation for the class to focus on DNA as a starting point. And so you see, you have the coursework listed here, and you have exams or quiz listed on the right-hand side. Then DNA structure and chromosomes, which is a very important chapter. It's a small chapter, which tells you, we know DNA. DNA is double helix made up of 4 bases, nucleotides, phosphodiester bond, purines, pyrimidines. But beyond that, we have to now look at the proteins that encapsulate DNA. And DNA plus protein, it makes a composite chromosomes, right? So we are going to study environment, um, environment around the DNA. And then you have quiz 2. Then you have, then we read about replication and DNA and repair. That will be quiz 3. After finishing transcription and translation, we will have test number 1, test 1. So while all the quizzes combined will be around 10 to 15%, test 1 by itself will be 15%. So, test all these tests are differing from quizzes based upon the weightage of percentage points. Then you have then regulation of gene expression, mutations, and cancer, cell cycle, and then you have test 2. Then there is quiz 5, and then you have test 3 and final. Sometimes students, they ask me to, for various reasons, drop one quiz or add one more quiz. So that part I'm flexible. But this is generally the structure of this course, starting from history, first the DNA, then DNA function, then transcription, regulation. So it's very systematic. It builds one above the other. They are not disjointed. So it's together. So this is in majority the syllabus. That's very important that I talk to you about the policies, right? Policies are very, very important in today's today's teaching setting. Just so number 1 is that how you email me, right? It's very important. So how do we communicate, right? We are not meeting every week. I'm not on campus. Sometimes students, they come to me during my office hours. Sometimes, I try to mediate with the students if the student is working, or what time it suits, so I can plan. But this is excuse me, this is totally online. So communication is very important. So number 1, I encourage you to ask me question and attend my office hours, which is thrice a week. If you choose to communicate via email, I'm teaching 3 classes, 461, 211, and Bio 499. So make sure in your subject line you write which class you are enrolled into. I generally get a lot of emails, and plus I do many things in addition to teaching. So keep it very professional, the emails. And there's always chances from email because everybody is using different platform. I encourage you to use school ID. And I have seen people emailing me through Gmail or Yahoo. Sometimes it doesn't reach me. CUNY has very different filter system at different time points. So which email would be, which will be, not given access, I don't know. I have no control over that. So if you email me and you don't hear from me in 24, 48 hours, best is to attend office hours or send me a reminder email. And I try to respond to students' email, on a very regular basis, provided the email comes to me. Secondly, I already mentioned that if you're attending the class, you don't need to put your camera on. You don't need to put your picture ID. So, this is a digital world. This is a virtual world. Although we are using it, we don't control it, right? So make sure I protect myself and you protect yourself. Then and also, so first quiz, second quiz, there could be many unexpected problems, right, with Blackboard. But Blackboard is mainly managed by the school or CUNY. If there are problems, then you need to contact the Blackboard representative in the school. Or if there is a problem with something you're trying to access, then you contact the school IT department, right? So I will try to help as much as possible with regard, but my knowledge of Blackboard is very limited, and I'm very honest about it. Why it is limited? That I use it for uploading my lectures, my videos, my recordings. I don't know everything about the Blackboard. Blackboard is a universe, very big. And I'm also learning Blackboard. One thing is knowing Blackboard, one thing is using Blackboard. So most of the assignments that I give, I know, but I know it from my end. The configuration of Blackboard is different for instructor and different for students. Although it's an interface between me and you, but you have to realize that at my end, things are different. At your end, things are, I believe, a little bit different. So if you have any obstacles, any hurdles, right away, do let me know that you are having some issues. But the person to contact is IT department in the school to resolve these differences. Right? And also look at the video conferencing etiquettes, how to, so these are very simple policies. It's for me as well and for you as well, both, such that the learning becomes effective. And at any point of time, you have a feedback for me, if you have suggestion for me, email me or come and join on the live session. Right? So with this, I finish introduction of syllabus and course policies. So I got a question. Said, Professor, sometimes I receive messages late from Blackboard, which is the reason I'm just noticing the discussion forum. Will I have until today to submit? Yes. So let me go through this new system, how I'm going to. So thanks for asking this question. So what I am going to do, so you will see week by week, I will be posting learning activities. Right? These learning activities will be composed of my lecture. Lecture will compose of slides plus my recorded lecture. Then you will have an assignment. Then third, there will be assignment on discussion board. Fourth, there will be a quiz. And fifth, I will put, before I give the quiz, I will post the recorded version of assignment. So these are the 5 things, minimum 5 things that will be the weekly learning activity as we advance. So for discussion board, for assignment, there will always be a deadline of 1 week, right? One working week, that is Monday to Friday. And discussion board also, you will get a deadline for 1 week. So let's say if I posted something on the discussion board, look at the time when I posted, the deadline is for 1 week, right? So the semester, so I'm looking at the calendar, the semester started on 26th, right? 26th of August. So the deadline for discussion board would be until 3rd September, September 3rd. So you will have deadline to do your assignment, whether it's on discussion board or you need to upload something. And if you have to upload something, I will create a folder for submitting your assignment. So I hope I answered your questions. That's what I see on the Blackboard, on the chat. And anywhere you see lack of clarity, I have 3 live office hours one day. So Tuesday, Wednesday, Thursdays, every day. I have not kept Monday because Monday I'm teaching 499, which is senior seminar. I kept office hours for them. Monday for my senior seminar class. For Tuesday, Wednesday, and Thursdays at 3 point of time, I have kept these office hours for Bio 461 and Bio 211. Any more questions? So this first week assignment is very important, because if you don't do, I will not be able to mark that you enrolled into this class, right? So in our face-to-face class in a lecture theater, if you're present, I marked you present. But here, there is no, attendance is not mandatory, right? I can't take attendance, but yes, if you don't do the assignment, especially the first assignment, I can't mark you present. And so take those assignments for first few weeks very seriously. Don't miss the deadline. Any more questions?
Okay.
Yes.
Did you post the syllabus on Blackboard?
Yes, it's already there.
Oh, I don't see it.
Yeah, I'll check again.
It doesn't show on my.
I don't know. That's what I'm saying, that you have to consult if you're not seeing something. Does anybody can anybody tell me? Did he or she saw the syllabus on the Blackboard? Did you see this? Did you see the syllabus on the Blackboard, class? Yeah, one of the students wrote on the chat that yes, the students saw the syllabus. So it's there. There.
It's there. Did you press content?
I see it now.
Okay.
Yeah.
Thank you.
Any other questions?
Did you say when is the next time we're meeting or no?
No, I can't host a class. I will post all the teaching material on the Blackboard as learning activities week-wise. But yes, I am available live during office hours. That is on Tuesdays, Wednesdays, and Thursdays. Look at the timing on the syllabus.
Okay.
So I'm not allowed to give a time that, okay, I'm teaching 10 to 11, so you can be here. No. Because there are some students who can say why you are teaching when we cannot attend. So best is office hours. So listen to the recordings, ask questions. That's what I'm here for. And I taught same way, Bio 211 in summer 2, totally asynchronous. So it was pretty successful. Students liked the flexibility part of it. So when I got the questions, so are saying there will be no more. I know I said that Zoom meeting is live, office hours, thrice a week. But this one, I was discussing the syllabus. So I said this is a non-mandatory asynchronous meeting. So this meeting was also optional. So going back to the definition of the asynchronous, so I want to follow that path. And if you're listening to my recorded lectures, please come back during my office hours. And give yourself a little bit time, just 1 week, see how things flow, and then let me know what more is needed from my end to make this course more effective. Because I have taught summer as asynchronous, which is very tight as compared to fall. I taught half of spring 2020 when we suddenly went to, but this is the first. So spring 2020 was synchronous after everybody had to be there on time. But this is asynchronous, which is first time for me, first time for you. So share your experience so that I can, if need be, I can be more flexible. So for instance, my Bio 211 class, they do presentations. So they can do presentations on either on live, or they can do upload their presentations on discussion board. So that's what. So if there are no questions, then we could end this meeting. And if anybody wants to talk to me, then that person could stay behind and we can talk together. So nice meeting you all.
Bye, Professor. We can leave, right?
Yes, yes. You could leave.
Okay.
Professor,
Yes.
I just want to clarify one more time. So everything is going to be asynchronous and we could just speak to you during office hours, right?
Yes.
Okay. Thank you so much.
Welcome. Any question? So if there are no questions, then I am ending this meeting.